Digestion of carbohydrates begins in the mouth. Mechanical digestion of carbohydrates, starches, sugars, and fibers found in many different foods begins when the food is chewed into smaller pieces. Chemical digestion begins when salivary amylase, an enzyme secreted by the salivary glands, begins to break down the food by splitting carbohydrate bonds with water. The food is then swallowed and moves through the esophagus. Salivary amylase continues to break bonds, traveling along in solution with the carbohydrates, or carbs.
When the carbohydrates reach the stomach, they are still somewhat intact, and the salivary amylase enzyme is inactivated in the stomach due to increased acidity. The stomach secretes acid, which aids further digestion. The carbohydrates pass through the stomach, and digestion continues when it reaches the first part of the small intestine, the duodenum. In the duodenum, the carbohydrates meet pancreatic amylase, another enzyme which comes from the pancreas and continues the process of breaking down the carbs.
Remaining sugars are processed by the brush border enzymes, produced by the microvilli of the small intestine. The larger surface area of the brush border, the many tiny projections sticking out of the small intestine, and the enzymes it secretes helps to increase nutrient absorption. Here, the carbohydrate molecules have been hydrolyzed, broken down, into their simplest form of monosaccharides that can be absorbed into the bloodstream.
Digested sugars then move into the microvilli of epithelial cells of the small intestine and enter capillaries, small branching blood vessels found in the small intestine. Absorbed substances that originated in the carbohydrate food are now in the bloodstream and can travel around the body to provide energy.
What can't be broken down and absorbed is carried through the large intestine, which absorbs water and sends the carbohydrates into the rectum. With excess buildup and pressure, a signal is sent to the brain to induce a bowel movement. The waste leaves the body in the form of feces.
Protein, a molecule made up of building blocks of amino acids. Digestion of protein begins in the mouth as well. Mechanical digestion of the protein begins with the chewing of the food, breaking it into smaller pieces. Once the food is chewed, it is swallowed and sent down the esophagus. Chemical digestion begins in the stomach with pepsin, the protein digesting enzyme secreted there. Pepsin breaks down the peptide bonds which hold amino acids together. When these are broken up, polypeptides, long chains of peptides, are formed and move into the small intestines. Additional enzymes now complete the digesting process.
In the small intestine, trypsin, chymotrypsin, and carboxypeptidase, digestive enzymes that come from the pancreas, work to break down polypeptides. These enzymes enter the duodenum, the first part of the small intestine, from the pancreatic duct. In the small intestine, the brush border enzymes, the many tiny projections sticking out of the small intestine on the microvilli, complete digestion. Here, polypeptides continue to break down, forming peptides, which are two or more amino acids linked together.
Polypeptides and peptides continue to break down until they are in the form of amino acids. Amino acids are small enough to be absorbed through the small intestine lining and then sent into the bloodstream. What isn't absorbed and broken down inside the small intestine moves to the large intestine, where the water from indigestible protein is absorbed. The leftover protein is transported to the rectum. As stool and excess protein build up, the outer walls of the rectum feel pressure and send a signal to your brain that you need to induce a bowel movement. In the form of feces, it leaves the body as waste. Lipids are fat molecules. They can't dissolve in water. First, lipids enter the mouth as food. Mechanical digestion begins as you chew your food and break it into smaller pieces. The lipids are then pushed through the esophagus but clump together because they have the inability to dissolve in water. The lipids proceed to move through the stomach intact. When the lipid clumps reach the small intestine, they have not been digested at all. The clumps begin breaking down by chemical digestion when bile, a salty liquid created in the liver and stored in the gallbladder, mixes with the fat globules. Bile salts within the bile help break the large fat clumps into smaller droplets and coats them. These broken up fat droplets now have a larger surface area.
Pancreatic lipase, a fat-digesting enzyme that is secreted into the duodenum through the pancreas, can only act on the surface area of fat droplets. It makes the smaller globs more useful because they have a larger surface area. Pancreatic lipase breaks down the fat droplets into free fatty acids and monoglycerides, the digestive products of lipids. Unlike with carbohydrates and proteins, the trip for lipids into the bloodstream is much more complex. Monoglycerides and free fatty acids have the ability to move out of the digestive tract and go through the process of absorption in the mucus-producing lining of the small intestine, called the mucosa.
The monoglycerides and free fatty acids enter the epithelial cells by passing through the mucosa. Inside the epithelial cells, the monoglycerides and free fatty acids enter the endoplasmic reticulum, which synthesizes and transports the lipids. Triglycerides are also formed. Triglycerides are coated with protein and form chylomicrons, which are protein-covered fats. Chylomicrons can now travel outside the cell due to the protein-formed water-soluble coat. The chylomicrons enter the lacteal vessels located in the villi, the many tiny tube-like structures that transport lipids through the lymphatic system and enter the bloodstream. The leftover lipids are transported to the rectum.
As stool and excess lipids build up, the outer walls of the rectum feel pressure and send a signal to your brain that you need to induce a bowel movement. In the form of feces, it leaves the body as waste.